Okay, so what if we could rewrite the code of life? You may have heard of genes, but have you ever thought of how these genes can be altered to improve life and make things easy? Well, welcome to our today's lecture on genetic engineering. So, genetic engineering is also called genetic manipulation or genetic modification. Okay, so what is genetic engineering? So, genetic engineering is the direct manipulation of an organism's DNA using biotechnology to alter its traits or functions. It enables scientists to add, delete, or change specific genes, even transferring traits across different species, to produce desired characteristics like disease resistance or the ability to manufacture medications. So the first recombinant DNA molecule was designed in 1972 by Paul Berg. So new DNA can be obtained either by isolating and copying the genetic material of interest using recombinant DNA methods by artificially synthesizing the DNA. So what is the process of recombinant DNA technology or genetic engineering? So the first process is identification of desired gene or trait. So you have to first ask yourself, what do I want to clone? What gene of interest or phenotype or characteristics am I looking for? So you can identify the gene through, we have technologies which have been developed like microarray, transcriptomics, or genome sequencing. So after you do that, you can now isolate your gene of interest. Then we move on to the next process, which is molecular cloning. So during molecular cloning, the gene is opened, and once the gene is opened, we are going to have DNA being DNA. So we go to DNA purification. Then after DNA is purified, we are going to have restriction enzymes, which are going to cut the specific sequence of your DNA. Then we are going to use a PCR to amplify the gene. This one you can use technologies like the gel electrophoresis can be used to now extract the gene. And then after that we are going to have insertion of DNA into the host genome. So how do we insert? So after we have extracted the gene, we are going to have ligation of that gene into a plasmid. So after the gene is ligated into a plasmid, the plasmid is inserted into bacterium, and from the bacterium we are going to have it being replicated into the bacterium. Okay, so what happens during inserting this DNA into the host genome? So before DNA is inserted into target organism, it must be combined with other genetic elements. So these are like the promoter or the terminator region. So the promoter is going to be to initiate the transcription, and the terminator is going to end the transcription. So we are going also to have the addition of a selectable marker gene. So in most cases, we usually have the marker gene being mostly one which has antibiotic resistance or confers antibiotic resistance. So we are going to have natural uptake of DNA, or we can also have the use of vectors. So a vector is going to be used to insert the DNA into the host genome. Sometimes the genome can also be inserted through the cell's nucleus using the nuclear envelope. So what happens during genetic engineering? So we are going to have plant genome are usually engineered using the agrobacterium, and agrobacterium is used to deliver the sequences which are hosted by the tDNA binary vectors. And these tDNA binary vectors are going to be inserted using agrobacterium mediated transformation. So how do we confirm if that new gene has been, we have successfully cloned, or we have successfully taken up the gene? So we are going to have further testing, which can be done either using the PCR or techniques like the southern hybridization, DNA sequencing, and for the RNA, we are going to have the northern blotting or the northern hybridization, western blot, ELISA, and or the immunofluorescence technique. And also sometimes you can have the real-time PCR. Okay, so this is a diagrammatic representation of what I was talking about. So from that diagram, you could see that the process from the DNA isolation to purification, to ligation, and then to gel electrophoresis, and then we are going to have the cloning, uh, confirmation by the Western blotting technologies. So what are the applications of the genetic engineering? So genetic engineering can be used in medicine, for example, in the manufacture of drugs, creation of animal models, like for example the mice, and gene therapy. So when you talk about manufacture of drugs, we have seen the manufacture of growth hormones, uh, hormones like the insulin being manufactured for people who are diabetic. Then in the creation of animal models, we have seen the use of the mice, which has human characteristics. So from the mice, you can study the genetic characteristics of a disease and how it will react if it was injected into a human subject. Then through gene therapy, it's where we have a defective gene being removed and replaced with a gene which is effective. So the next one is in research. So where we have organisms are being genetically engineered uh, to study to study diseases or the function of certain genes. Then in agriculture, we are going to have genetically modified crops, and in industry, for example, in the industrial microbiology, where we have for example, mass production of protein. So we can grow organisms in bioreactors using industrial fermentation and then purifying the protein. So is genetic modification. Once we do this genetic modification, what happens? So we are going to have genetic modification. Different countries have come up with ways through which they can regulate the genes or the genome, or what the GMO, that is the genetically modified organisms or plants or crops. So when we talk about regulation, we talk about the approaches taken by governments to assess and manage risks associated with development of the GMOs. And with this, thank you for watching.